afternoon. I'm Mark Carther. I'm a professor of medicine and pathology and molecular medicine at McMaster University, and today at the American Heart Association meeting, I presented the final results of our phase 3 study looking at the use of andexanet alfa for the reversal of the direct oral anticoagulant rivaroxaban. This presentation was part of an ongoing program of research which is in the process of developing the first specific reversal agent for the 10A inhibitors. As many of you know, anti-10A inhibitors such as rivaroxaban or apixaban are being used with increasing frequency to reduce the risk of thrombotic complications in patients such as those with atrial fibrillation or those with deep vein thrombosis or pulmonary embolism. Although bleeding with these agents is both less severe, less common, and less likely to cause death than bleeding with the traditional oral anticoagulant warfarin, there still is a need for a specific reversal agent to antagonize the effect of these drugs, both because patients are at risk of bleeding, but also because clinicians identify the lack of a reversal agent as a reason why they are hesitant to use these drugs. From our perspective, andexanet is moving forward at high speed and towards, I think, a successful outcome in terms of its approval. What we presented today was the results showing that for the drug known as rivaroxaban, the use of a bolus of 800 milligrams followed by an infusion of 960 milligrams of andexanet over 2 hours is able to almost instantaneously and almost completely reverse the blood thinning effect of rivaroxaban, producing normal hemostasis as measured by a test known as thrombin generation. Currently, we're proposing and currently enrolling patients in a phase 3b/4 study, which is enrolling patients with active bleeding and treating them with andexanet to see whether or not we both can prove reversal using a lab test known as the anti-10A level. From my perspective, this drug is very, very important, as I said earlier, partly because it should directly reduce the consequences of bleeding in patients who do have that complication, but probably more importantly because it'll provide clinicians with reassurance that it is safe to use those drugs, particularly those patients who have a higher risk of bleeding or those patients in whom clinicians are a little bit afraid to use anticoagulants. They know the drugs will reduce their risk of, for example, stroke in the setting of atrial fibrillation, but are fearful that if they cause bleeding, there won't be an effective way to reverse it. So in summary, the andexanet program, the final study for which we presented today and which was published today in the New England Journal, provides the background confirmation of the effectiveness of andexanet alfa. The next phase, which we're well embarked on at this point, is a clinical trial showing that that drug is effective both in terms of its ability to reverse the blood thinning effect of these drugs in actively bleeding patients and in terms of its ability to improve hemostasis as measured by the clinicians who are caring for those patients. Thank you.